The weekend is over. And Monday's almost over. But first, there must be biochemistry. That's good, right? Had a good weekend? I spent a fair percentage of the weekend getting the videos done. I finally got through those, and they're all caught up, and I dealt with the technical issues that I was having. So I think it's smooth sailing here forward, which is good news. Speaking of video, today, filming the class and parts of the class will be Daryl Lai, who is back in the back, and he'll be walking around the room a little bit, taking some video. If there's any reason that you would not like to be in that video, please let me know at the end of class, and we'll make sure that you don't make it into the final version. So you don't have to be in it. There's no requirement of any sort. So if you would like not to be in the video, please let me know at the end, okay? Okay. So last time we got through a fair amount of discussion about the beginning structure of proteins. We're going to spend a fair amount more time talking about structure of proteins because, as I've been alluding to, the structure of a protein is the most important consideration we have for it. When we lose its structure, we lose its function. You might say, well, what's the structure? Well, a protein has a very specific shape. The way in which all the amino acids interact with each other is very essential for the protein's function. If we disrupt that, we lose the function. And we're going to see more about that as we get going along today. When I finished last time, I was just getting started talking about peptide bonds. And so I want to go back to that, and that's where we'll start today. So as I mentioned last time, peptide bonds are, of course, the links between the building blocks of proteins. We make them by joining the amino end of one amino acid to the carboxy terminus of the other. So there's, in this case, an alpha carboxy right there. There's an alpha amino. And we can see these join together. They split out water. And the result is a peptide bond. A peptide bond is a covalent bond. Covalent bonds, you remember from the discussion of bonds, are very, very strong bonds. It takes a lot of energy to break peptide bonds. Okay? And so that's pretty important because we don't want the protein to fall apart. When we think about other structures, like I talked about hydrogen bonds, which are much weaker, okay, we will see later that they also play a role in overall protein structure, but not in joining individual amino acids together. They may have regions, so one region of a protein may be attracted to another through hydrogen bonds, and that gives some structure. But for our purposes right now, peptide bonds are the most important thing in terms of strength holding together a protein. Okay. Well, if we take more than two amino acids and put them together, we start making a polypeptide. Okay. Now, there actually is a distinction between the term polypeptide and protein, but it's really more esoteric than we need to be concerned with here. So I will use the terms polypeptide and proteins interchangeably. Okay? I will use those terms interchangeably. This is a nice schematic, and I like it because it shows all the various peptide bonds that are in this polypeptide. Here is the amino acid at the N-terminus, and we call it the N-terminus because it's at this end of the protein. That's the only free alpha amino group. The other end is called the carboxy terminus, or the C-terminus, and it's the only place in the protein we have a free alpha carboxyl group. Now, we do have, within the protein, we'll have some R groups that'll have carboxyls and aminos that'll be free, okay? But the only free alpha amino and the only free alpha carboxyl are at the ends of a polypeptide. So we can tell one end from the other. People talk about the N-terminus versus the C-terminus, meaning the amino end versus the carboxyl end of the polypeptide chain. Okay. Now, another thing I want to talk about with respect to peptide bonds is something I'll talk about briefly today. I'm not going to go into much depth about it, but it's kind of interesting. All right? And this is the fact that these peptide bonds that we can see here, they're drawn as single bonds. And if you remember your organic chemistry, you should remember that single bonds can rotate. Okay? So you can freely rotate around a single bond, and we can imagine a lot of rotations that would happen through all of these single bonds that are on here. The peptide bond is not truly a fully single bond. It has what we call a resonant structure, which means that sometimes it has the characteristics of a double bond, and sometimes, excuse me, it has the characteristics of a single bond. Okay? Now, because of that, this guy we can treat more like a double bond than we treat like a single bond, meaning that it doesn't really rotate. It doesn't really rotate. Okay? Now, what that means, if you recall from your organic chemistry, is that something that has a double bond, because it doesn't rotate, it makes a planar structure, flat. Okay? When we look at this, we see that there's a flat structure. There's a flat structure over here. Okay? So there's one and there's another one. In between there are two different single bonds. These guys actually are single bonds because they're not peptide bonds right here. All right? Double bond, double bond. These are each single bonds. Okay? Now, what that means is that we can think of these double bonds as defining a plane. That is a flat surface. This flat surface is going to have R group coming off of it. It's going to have a hydrogen coming off of it, etc. Right? So here's one of those bonds over here that's flat with the peptide bond. Here's the other one over here. And in between, we've got a carbon that's joined by two single bonds. Okay? The carbon, we can imagine, is between my two thumbs. Okay? Those bonds around that carbon are freely rotatable. Okay? Whereas these bonds in my hand don't rotate. Okay? The whole plane has to rotate. We don't see something within my hand rotating. All right? Now, that's important because we know from both theoretical studies and actual studies that there are limitations in the angles that those single bonds can rotate. You might think, well, they can just freely rotate all the way around, but it turns out that they can't. And they can't because what I'm trying to show you with my fingers is that there's steric hindrance. They start bumping into each other in certain angles. These are big groups hanging off of here, and they can bump into each other. Okay? The person who discovered this was an Indian scientist named Ramachandran. Okay? Ramachandran predicted accurately that in fact this was the case. And he said we could look at a polypeptide in terms of structure is just the way I'm telling you right here. A flat structure, a carbon in between where my nose is, okay, and singles like this. All right? So the single bonds can rotate, but that means the whole plane of this rotates and these planes bump into each other. So he predicted that there would be certain angles that would be unstable. Okay? He was dead on. We know that that was the case. And he actually used a theoretical plot we call a Ramachandran plot to predict that. Now, I'll show you one a little bit later, but I'm just building up to this because I've got this structure on the screen to show you right now. When we look at a Ramachandran plot, what we see are all the possible angles for one of the bonds and all the possible angles for the other bonds. So, theoretically, one single bond could rotate through all of 360 degrees, and theoretically, the other one could rotate all the way through 360 degrees. Okay? So we see a plot of from a 180 down to a minus 180, from 180 up to a minus 180, and we see on this screen, we see on the plot, regions of stability. Okay? Turns out there's two major regions of stability in a Ramachandran plot. And those correspond to some structures we're going to talk about in a little bit. They were structures that were actually discovered by Linus Pauling. Okay? So, questions about that? I know it's a little hard to envision in your heads as I'm going through that. Can you picture it? I'm not going to make you draw one on the exam. But it's still helpful to picture it. It's still helpful to picture it. Okay. All right. So, and I'll come back and say a little bit more about that in a bit. Now, here's the planar structure. This is, again, reiterating what I'm telling you here. I said this peptide bond has a resonant structure, and that means it actually borrows some of the electrons from this double-bonded oxygen and makes a double-bonded structure like we see here part of the time. And that's what gives it its double-bonded nature. Yes, sir?
So would you use like a name plot to figure out where this gouge lives? Is that...
We can find out exactly where the interactions happen that will make it unstable, essentially. Yeah, you bet. Yep. Is something you should know?
Yeah.
Unless I talk about it? All right. All right. Now, So what I'm going to do now is, your book is, I shouldn't say your book, but the way I've got this organized is a little odd in the sense that I've got a couple things to talk about unusual amino acids, and then we're going to go talk about more elaborate protein structure. What we see in protein structure is that protein structure we can describe at 4 levels. Okay? We're getting ready to talk about the first one. So you've got the background now, you've got the peptide bonds, you've got the amino acids, and we're going to dive into the 4 levels of protein structure in just a bit. Okay. Well, there are some interesting amino acids that relate to human health that you should be aware of. One is histamine, as you can see here. I'm sure everybody who's ever had a cold has heard the term histamine, only they've heard it in the terms of antihistamine, right? So antihistamine is a drug that you take to stop the production of histamines, and histamines are what we associate with congestion, okay? So snot and all the various lovely things that go with that. By stopping the production of histamines, we do tend to dry out. Histamines are produced from the amino acid histidine. Okay. Another one that you will commonly, especially for nutritionists, that you will hear something about is aspartame. You've heard of aspartame before. You probably know it as nutrasweet. Aspartame is a very simple dipeptide that is made by joining together 2 amino acids. One of the amino acids is phenylalanine. That's the ring that you see up over here. Okay? I tell you that because aspartame is, in many ways, one of the tamer artificial sweeteners that's out there. Okay? Not a lot of side effects. There are in large quantities, as there are of anything else. But in small quantities, it's really not too much of a problem, unless you have a disease known as phenylketonuria. And by the way, I'm not going to spell these out. I'll put them in the highlights. You can see them there. Phenylketonuria is a disease where people lack an enzyme that allows them to break down phenylalanine. If they can't break down phenylalanine, they have very serious side effects with that. So people who have phenylketonuria should never drink something that has NutraSweet in it because they will suffer some very dire consequences. They have to be very careful with how much phenylalanine they get into their diets. Okay. These last guys I'm going to let go here, and we'll talk about the protein structure in 3D. Okay, so as I promised, we're going to study protein structure very up close and personal, and we're going to look at it at 4 different levels. All right, they're known as primary, secondary, tertiary, and quaternary. Okay. Each of these steps back another step in looking at a protein structure. The closest in will be the primary. Secondary, we step back just a little bit. Tertiary, we step back further. And quaternary, we step back even further. They each have distinctive qualities, and they each are responsible for giving proteins the overall, what's the word I want? Overall activity that a protein has. Okay. Well, I talked about Ramachandran plots earlier. Now I'm going to show you how the Ramachandran plots are actually done. Okay. So this picture on the screen shows you those 2 planes I talk about that correspond to the peptide bonds. There's one plane there in green. Here's another plane here in green. And there's that alpha carbon in the middle joined by 2 single bonds. Okay? The angles that can be rotated around those single bonds have names. They're called phi and psi. And no, you don't need to know which one is which. I don't care that you know that. But phi and psi angles are rotational angles. That is the rotating of that sphere, of that plane. Okay? And there are certain rotational angles that we will see are stable, and certain rotational angles that are completely unstable. All right. Linus Pauling won the Nobel Prize. He's the most famous scientist ever to graduate from OSU, and probably the most famous scientist to graduate from anywhere in the world, for that matter, in the 20th century. One of the 2 Nobel Prizes that Linus Pauling won was for chemistry, and it was specifically for the chemistry involved in the structure of proteins. Okay? Linus Pauling discovered 2 structures that turned out later in a Ramachandran plot. One of which is known as an alpha helix, The other of which is known as a beta strand. Okay? A beta strand. Okay. What you see on the screen is a depiction of an alpha helix. We're actually looking at a protein very up close and personal. Okay? An alpha helix is a coil. And that coil arises as a result of two things. One, the structure of the amino acids that are comprising that coil. And the second corresponds to the hydrogen bonds between them. Hydrogen bonds. So now I said something about, well, it matters upon which of the amino acids comprise this structure that you see here. You can see, and by the way, that green that's there is just to guide your eyes to see the helical nature. It's not a real thing. Okay? The actual atoms that you see are all shown there, but the green is to help your eyes follow the helical nature of this polypeptide. Okay? So I said it matters what comprises the amino acid, what the amino acids are in there. Okay? It tells us that some amino acids will give rise to a structure like this if they're present in the sequence of a protein, and others will not. Others will not. Now I'm going to talk about that in a little bit more depth here. Well, why would some amino acids lend themselves to this and others will not? I want you to look at that structure that you see on the screen, and when you look at that, you can say, whoa, okay, that's pretty crowded in there. That's pretty darn crowded because there's a lot of atoms in there. And this guy is coiled upon itself. Okay? We might imagine that if we had a protein that had, let's say at this point, an R group that was rather large and bulky, it might in some way interfere with the structure here because it just wouldn't fit. In fact, that turns out exactly to be the case. Turns out exactly to be the case. Frequently, we will see amino acids present that will just basically disrupt a structure from occurring. Okay? When that happens, we typically in a protein will get a bend, we'll get a coil, and then we'll get a bend, and then we'll go off and make something else. And we'll see that in a little bit. What I'm describing here are the first two levels of protein structure. Okay? You're getting two at once. The first one is the primary, and that corresponds to the sequence of the amino acids. The sequence, that is, starting at the N-terminus, I might have a methionine attached to a serine, attached to an aspartic acid, attached to a glycine, etc. That's a sequence. That's a specific sequence of amino acids. The sequence of amino acids corresponds to the primary structure. That's as up close and personal as we're going to get. Because of that sequence, certain portions of a protein will form coils, like this guy has done. This coil happens because the sequence of amino acids that were in this region of the protein did not have anything that bent it out of a double helix. So it stayed in a double helix. Okay? This that we see on the screen is the secondary structure. So a double helix is an example of a secondary structure. Now, secondary structure, you can see, arises from interactions between amino acids. And in fact, these interactions are shown by these little yellow guys right here. You see the little yellow things right there on each one? Those are hydrogen bonds. Hydrogen bonds are helping to stabilize this alpha helix. Okay? So an alpha helix is stabilized by hydrogen bonds. Now let's think about this. What did I say about hydrogen bonds relative to covalent bonds? Weaker. Okay? So we might expect then it would be much easier to disrupt an alpha helix with a little bit of energy than it would be to pull apart the amino acids in the primary structure, right? It tells us that primary structure will be very stable because of the covalent bonds in it. This guy is going to be less stable because hydrogen bonds aren't as strong as covalent bonds. And it turns out that's exactly the case. All right? I've been telling you that structure matters for a protein. This structure within a protein matters a lot. If I disrupt this structure, I'm going to change the overall structure of the protein. And as a consequence of doing that, this protein may likely lose its function. All of you nutritionists should make note of this. This is why we cook our food. We cook our food to disrupt protein structure. We can use the heat, we can use boiling water, for example, and that will break the bonds in alpha helices. Alpha helices will come apart. Protein structure will fall apart. When a cell loses its proteins, it's hosed. It's dead. We cook our food to kill the proteins, to kill the cells. Okay? All right. This is one of two secondary structures I'm going to talk about here. You can see this in a couple different ways. I like this one. Okay? This is a nice depiction of an alpha helix in front of Linus Pauling's childhood home. Kind of a cool thing. Okay? This is where he grew up, and they've erected this structure there, which I thought was kind of cool. Okay, and this I also like, and this was Linus Pauling's original notes about what protein structure might look like. Okay, this was how he drew an alpha helix. He didn't draw the coil on here, but he had the coil in his three-dimensional mind, but writing out how everything would look, how everything would fit in a protein when he predicted correctly the structure of proteins originally. First drawn in March of 1948. Okay. Now, there's another structure that Linus Pauling discovered that is in proteins, very common in proteins, that we need to talk about. I call them beta strands. All right? Many people refer to them as beta sheets, but technically that's not right. Okay? Technically, a beta sheet is comprised of a bunch of interacting beta strands. Okay? What is a beta strand? A beta strand is like a very simple but kinky helix. A helix actually coils, right? You know what a coil is like. A spring is like a coil. It coils. It goes around and round. A beta strand doesn't do that. It does it in 2 dimensions instead of going in 3. So here's the 3 dimensions where it's going up. A beta strand will go doink, 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 doink. Okay? It's like the pleats of a curtain. Okay? They're angular. They're not twisted around like this. They are angular. They're 2-dimensional structures. Okay? That's what we see in a beta strand. Here's one beta strand here on the left. And you can see it's interacting with another beta strand on the right. And when we put more than one of them together, we make a sheet. That's why they're calling this a sheet. One of them is a strand. More than one is a sheet. Okay? Now look at what's holding the beta strands together into the sheet. Hydrogen bonds. Hydrogen bonds. Hydrogen bonds. Okay? And just like we can disrupt the structure of an alpha helix with heat, so too can we disrupt the structure of beta strands with heat, because hydrogen bonds can readily be broken with the heat that we might use to cook food. Okay? Now, proteins will have, in many cases, both alpha helices and beta strands. Some proteins we're going to see will have primarily alpha. Some will have primarily beta. We don't need to worry too much about this. But the fact is that these structures that we see commonly occur in many, many proteins. Okay? They occur in many, many proteins. All right. That's a depiction of a beta sheet. And you can see here's one end of the protein starting over here. You can follow the arrows. Look how it's going up. And now this is a region that is a beta strand interacting with another beta strand, interacting with another, etc. Notice how the beta strands are organized with respect to each other. Okay? We see these bends. These bends might predict, based on what I told you about alpha helices, occur when there are amino acids that wouldn't be conducive to making a beta strand. And that's exactly right. These bends will have amino acids that aren't very conducive to making beta strands. Okay? Kind of cool stuff. Okay. Questions about that? You guys are back. I think Mondays are the quiet group for this class. I don't know. Last Monday was very quiet. The rest of the week, you guys seem to be alive. But Mondays, maybe Monday afternoon at 4 o'clock, things just don't fly very well. Okay. Well, what about those turns? What about those turns that I talked about? So I said there are some amino acids that really don't give very good alpha helices or very good beta strands. Okay? Well, turns turn out to be very important in the overall structure of a protein. Okay? They turn out to be very important because the protein, as we will see, most proteins go through a phenomenon called folding. Folding gives a protein, most proteins, its shape. Okay? That involves another level of structure I'll be talking about in a minute. But folding happens thanks to the fact that we have bends that occur in the sequence of the protein. All right? So where are they? Well, we can see here's a nice bend here. Here's a nice bend here. Wherever we see these little white things here that aren't in a coil of some sort, those are all corresponding to bends. Okay? Some amino acids in a protein sequence are very conducive to making bends. One is one I talked about on Friday. It's proline. Anybody remember why proline was of interest? No, that's glycine. Nice try, though. It actually has the R group comes back around and it bonds with the nitrogen. And I said when that happens, it can't rotate anymore. It can't rotate because it's in a ring structure. The R group comes back and attaches itself to the alpha amino group on the amino acid. It's the only one that does that. As a consequence, proline is not very flexible. One of the things that's important for protein structure is flexibility. We're going to see as we get talking further about this that slight movements and slight flexibility within protein is really critical for a protein's ability to catalyze a reaction, for example. Proline has little flexibility. And so it's what you see is what you get. So it pretty much, when it's put into a protein, it's got a fairly rigid structure, and in many cases will actually cause a bend to occur where it pops in the structure. It's not always, but in many cases it will have a bend where it appears in the structure. Okay? So proline is something we would really expect would disrupt an alpha helix. It might very well disrupt beta strands, etc. Some of the bulkier amino acids like tryptophan or phenylalanine may also, because of the large R groups, also be not very consistent with the needs for the structure for an alpha helix or a beta strand. Okay. Some of these are really beautiful. I like this one. Okay. Okay. Draw that on the first exam, right? So, you know, I mean, this is what we call a beta barrel. Okay. A beta barrel, as its name suggests, is something that has beta strands, and look how they're arranged. It's really beautiful, I think, how they're arranged. And in the center of that is actually literally an opening. It's a barrel. Okay? Enzymes use barrels to do a variety of things, but one of the things that barrels are sometimes used to do is protect things on the inside. Protect things on the inside. Okay? We'll talk about, I won't go through some examples here. I'll talk about some of those later. Okay. Now, last time I briefly mentioned collagen, and if you remember what I said about collagen last time, it was that collagen was the most abundant the most abundant protein in your body. It was the connective tissue, the glue that holds you together. And I talked about how if you don't, covalently cross-link those individual strands of collagen, what happens is you get a very weak collagen, and as a consequence, you fall apart. And the reason that was important was because vitamin C is necessary to make those covalent bonds between the strands of collagen. When you don't have vitamin C, you get scurvy. And scurvy is literally means you're falling apart because your collagen is very, very weak as a consequence of that. Okay? Well, this shows one of the helices of collagen. Collagen is actually a triple helical structure. You see 3 different intertwined strands right there. And these intertwined strands are bundled. So we could see one strand here, we could see another one over here, we could see another one over here, we could see another one over here, etc. And then how those are all covalently linked to each other is what gives collagen its strength. Okay? One of the things that happens as you get older is your collagen gets older. Okay? I would wager to say I'm older than anybody in this room. Okay? And my collagen is much more cross-linked than yours is. What does that mean about my flexibility? It means that the older I get, the less flexible I will get. Okay? My collagen is getting old. Maybe I should stop taking vitamin C. That's what people always say. Okay? All right. Okay. Now, I'm going to show you something that's going to surprise you about collagen. And here it is. Okay? I'd like you to look at the things in boxes there. We see in the sort of yellowish boxes glycine appearing all through the thing. And we see proline, that's PRO, appearing there. Here we saw a helical structure. And I said proline was disrupting helical structures. Am I telling you lies? Well, it turns out that in collagen, proline does make helical structures. You also see HYP, that's hydroxyproline. That's where those hydroxyl groups get put on for the covalent bonding. We see a lot of proline. We see a lot of hydroxyproline inside of collagen. How in the world, based on what I've told you about lack of flexibility, does collagen form into a helix? Any thoughts based on what you know or what little you know about amino acids? I'm open to guesses here. I've actually told you the answer. The glycine plays a very important role. Okay? What do you guys remember about glycine? It has an H for an R group. It's got the smallest R group. And I said R groups can, in some cases, get in the way of things. What do you suppose is going to be the most flexible of the amino acids? That's going to be glycine. Okay? Glycine isn't just coincidentally next to proline or hydroxyproline through here. Glycine is there for a very important reason. It's there to allow that helix to form. Okay? Kind of cool stuff. Kind of cool stuff. Okay. And I showed you these before. Hydroxylysine, hydroxyproline. These are 2 different amino acids that get hydroxyl groups put onto them. And these are the ones that get joined together covalently later to give those cross links to give collagen its strength that it has. Okay. Now, talking about protein structure, there's one other thing I want to consider. All right? Collagen is an example of one class of protein. I'll give you some other examples in a minute, that we call fibrous proteins. Fibrous in the sense that they are literally fibers. Okay? They form a helical structure, and that helical structure just goes on and on and on and on and on and on. Okay? It doesn't stop. It just keeps going on and on. Your hair is a fibrous protein. Okay? It goes on and on and on. In my case, it doesn't go on as much as it goes on you guys, but it keeps going on and on and on and on. Right? Okay? Fibrous proteins have primary structure. And the helices that they have are secondary structure. But I'm getting ready to talk about tertiary structure. And one of the things we don't see very much of in fibrous proteins is tertiary structure. Okay? You'll see why I say that in a minute. All right? Fibrous proteins just basically have helices that go on and on and on and on. It could be alpha. It could be an alpha helix like what we have in our hair. Our hair is largely a protein that has alpha helical nature. It can be beta strands and beta sheets. Silk, for example, is a fibrous protein that is comprised mostly of a protein known as fibrin that has beta strands arranged in sheets. Okay? Both of these are considered fibrous proteins. Now, I said that fibrous proteins don't have tertiary structure, so now I need to tell you something about tertiary structure. Let's start by looking at this guy right here. We're going to talk more about it in some more detail later this week, actually. And it's hemoglobin. This guy doesn't look like a fibrous protein. That fibrous protein, you saw collagen just went on and on and on and on. No bends. Okay? This guy's full of bends. Okay? This is an example of what we call a globular protein. Globular proteins don't go on and on and on and on. They go on little ways. So we see a helix going for a ways, and then they have a bend, like we've been talking about. Then another helix goes on a ways, and it bends. And then it goes on a ways, and it bends. Okay? Bending gives tertiary structure. Tertiary structure arises from interactions. I'm going to give you a definition here. Okay? Tertiary structure arises from interactions between amino acids that are not close to each other in primary sequence. I'll repeat that. Tertiary structure arises from interactions between amino acids that are not close in primary structure. You say, well, what's not close mean? Basically, if they're more than 10 amino acids apart, that's not close. In an alpha helix, we saw interactions between amino acids that were pretty close in primary structure. They were all less than 10 amino acids apart. Okay? So primary structure, we've got peptide bonds. Secondary structure, we have regular repeats. They arise from hydrogen bonding that's occurring between amino acids that are close to each other, less than 10 apart. Tertiary arises from interactions between amino acids that are not close to each other. What does that mean? Well, look at the way this guy is folded up. Here might be amino acid number, right here might be amino acid number 51. And over here on this other chain might be amino acid number 130. And they might be forming between them a hydrogen bond of some sort, or some other kind of bond. Okay? So consequently, what folding does is it brings together regions that didn't start out close together. They didn't start out close together. That's what's happening during the process of folding. This guy starts out linear. It folds back up. It coils back up. Yes, question? Yes, so the question is why 10? Okay? And 10 is just sort of an arbitrary number. But when we look at a helix, for example, so if I start with a helix that goes, let's say, around this thing, wrapping around here. If I go one turn around for an alpha helix, that's 7 amino acids from here to here by the time it repeats itself. So if we say 10, we're not going to have anything like helical interactions that are happening. It's happening because regions have been brought closer together. Does that make sense? Okay. Now, the vast majority of proteins on the face of the earth are globular. The vast majority of proteins on the face of the earth are globular. Okay? Hair, nails are fibrous. Silk is fibrous. Okay? Collagen is fibrous. And it's very abundant in us. Okay? But in terms of overall proteins, the vast majority are globular. Well, so here's tertiary structure. Here's another look at a related protein known as myoglobin. I like this figure right here a lot. Okay? It shows, first of all, myoglobin, when we look at this, we see it's coils with a lot of bends. All the coils there are alpha helices. You don't see any beta strands in here. You don't see any kinky, bendy things like that. Everything in here is alpha helix. All right? But we see some other things. Look at this guy over here. This is a heme group. And you guys hear of hemoglobin? Hemoglobin gets its name by virtue of the fact that it has a heme group. Myoglobin is also an oxygen holding protein. And it also has a heme group right there. We'll see this in another angle in a little bit, but it's actually a flat planar structure, or pretty close to flat planar structure. And this red guy right down here is an oxygen. This oxygen is being held in place by this heme, and if we look right there above the red, you'll see another little red section right where my pointer is. That little red section right there is an atom of iron. Iron is holding onto the oxygen. It's actually the iron that binds to the oxygen. It's because of this iron is one of the reasons we need to have iron in our diet. So we can make a heme group, so we can make hemoglobin, so we can make myoglobin, so we can hold on to oxygen. Okay. But again, you can look here, alpha helix, bend, alpha helix, bend, alpha helix, bend, etc. And you start to see those bends play really important roles in determining the overall structure of a protein. Okay. Now, this structure, okay, schematically shows us all the different kinds of interactions that help to hold together a protein. Okay? Now, it doesn't show you covalent bonds, but I trust you know what peptide bonds look like because I'm not going up close and personal with that. But this shows several things that I think that you should recognize. First of all, here's a secondary structure. Okay? This secondary structure in this case is an alpha helix. We can see interactions, see the hydrogen bonds there in the red, hydrogen bonds in red. And as I said, in the case of an alpha helix, these amino acids are about 7 apart. They're not very far apart. This alpha helix has a bend and it goes to a beta strand. This particular beta strand is, in fact, a secondary structure. But when it's combined with something over here, we give rise to a tertiary structure because these guys are further away. All right. Hydrogen bonds are important in stabilizing secondary structure. Hydrogen bonds are also important in stabilizing tertiary structure. We can see here another thing helps to stabilize tertiary structure. And this is another covalent bond. It's known as a sulfhydryl bond. It's a disulfide bond. That shouldn't say sulfide, that should say disulfide. Okay? A disulfide bond arises between two sulfur atoms. Now we have 2 amino acids in our body that have sulfur in them. One is methionine. Methionine does not, underline this, does not make disulfide bonds. Okay? Does not make disulfide bonds. The other amino acid is known as cysteine, cysteine. Cysteine does participate in making double bonds. Cysteine has a side chain. It has an R group that has an SH. That's actually the sulfhydryl. And when you put two SHs together, you put them in very close proximity to each other, they will form a disulfide bond. And that's the SS bond that we see here. Now, that SS bond is a covalent bond. A disulfide bond is a covalent bond. Now, that's important because in tertiary structure, we see that there are, in some cases, covalent bonds that help to stabilize tertiary structure. Okay? This is being stabilized, this particular thing is being stabilized by that disulfide bond right there. There are other bonds that are important in the overall stability of a protein. I'll finish with this and then we'll have a song. How about that? Okay? Ionic bonds. Not too surprising. Pluses attract minuses. If we ionize a carboxyl on a side chain, we've got a COO minus. We ionize an amine on a side chain, we've got an NH3 plus. They can attract each other, and that can also give some stability to the overall structure of this protein. metal ion or metal atoms can also give some stability. They can be, they're not really covalent as such, but they can act like covalent bonds and actually give some stability to protein. This case I've drawn an iron atom that is helping to stabilize, holding together these chains into an approximate structure as we see here. The last structure, the last bonds I want to talk about to stabilize the structure of a protein are a little hard to understand at first. They're called hydrophobic interactions or hydrophobic bonds. Now these arise from interactions between amino acids that have hydrophobic side chains. Phenylalanine, isoleucine. If you look at the categories of amino acids I gave you, you'll see there's about 6 or 7 of them that I listed as being hydrophobic. Okay? What does this mean? These guys like each other. They like each other. And the thing that they have in common is that they all dislike water. Okay? If you put oil in water, what happens? Well, nothing. They stay apart because oil doesn't want to dissolve in water. They avoid each other. Within a protein, amino acids that don't like water will associate with each other so that they don't have to interact with water. Now this turns out to be a very cool property about proteins because what happens is that they will tend to be on the inside of a protein. They will fold and be on the inside, so they interact with each other and water is excluded. Many, many proteins, I would say probably most proteins, most globular proteins, have a central core that is fairly hydrophobic and an exterior surface that is fairly hydrophilic, meaning it likes water. Okay? Now, I know I've gone through a lot of stuff today. Before we sing a song, can I ask for any questions? Yes?
So thymine doesn't make disulfide bonds?
Methionine, yes.
Methionine, and then cysteine does.
Cysteine does. Okay? Okay, well, the song today doesn't cover all of protein structure, but it gives you a pretty good idea. We'll finish the rest of it on the next time. It's called A Little Protein Molecule. Oh, little protein molecule, you're lovely and serene, with 20 Zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats, arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. And tertiary structure gives polypeptide zing because of magic that occurs in protein folding. A folded enzyme's active and starts to catalyze when activators bind into its allosteric sites. Some other mechanisms control the enzyme rates by regulating synthesis and placement of phosphates. And all the regulation that's found inside of cells reminds the students learning it of pathways stray from hell. So here's how to remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. We'll talk about that later. See you guys next time.